So now let's take a look at the difference between sex linkage and autosomal linkage. Sex linkage simply refers to the genes that are on the sex chromosome, so either X or Y. Autosomal linkage. Autosomes, firstly, are chromosomes that are not sex chromosomes. Now, as we covered a little bit in the definition of linkage, autosomal linkage is where two genes will be positioned very closely to one another on the chromosome. And what this does is it links them. Now, during prophase 1 of meiosis 1, when we have crossing over, those two genes will tend to remain together. They will tend to not be separated. And so they will be linked. And so they will be inherited together in the next generation. Epistasis is where one gene at one locus affects the display of another gene at another locus. So, for example, we could have fur color, okay? Fur color in a mouse. Gene A codes for the color. Let's say gene A is coding for a black fur color. And then we have gene B. Gene B is coding for the dark pigment melanin. If gene B is not being produced properly and melanin isn't being produced properly, then it's going to be impossible to make that dark colored fur. So that is an example of epistasis, where although gene A is coding for a dark pigment, a gene at another locus is affecting the display of gene A. So now let's take a look at the chi-squared test. As we discussed in the test cross example that we did on the whiteboard previously, the expected results can be different from the observed results. And in a nutshell, this is because each fertilization is independent of any other fertilization. So the chi-squared test is used to see if the difference between the expected results and the observed results is significant or not. Is it due to chance, or is there actually possibly another factor that is having an effect on the observed results compared to the expected results? Now, in the calculation, it's important to understand what the difference between, or what the relationship between degrees of freedom and number of classes is. So number of classes simply means the number of categories. And degrees of freedom is the number of categories minus 1. Okay, so number of classes is the categories that we have. And then we go to degrees of freedom, and degrees of freedom is going to be categories take away 1. So now let's take a look at some recessive mutations that occur in humans, and we'll start off with the example of albinism. So albinism happens because of a mutation in the TYR gene. Now, the TYR gene codes for the enzyme called tyrosinase, and this enzyme hydroxylizes the amino acid tyrosine into DOPA. The tyrosinase enzyme then oxidizes the DOPA chemical, the DOPA molecule, into dopaquinone. And dopaquinone is used in the synthesis of the dark pigment melanin. So without a properly functioning tyrosinase enzyme, that dark pigment melanin cannot be produced. And that will result in a person in the phenotype being pale skin, white hair, and an iris that is almost transparent, and it will actually appear pink because the retina is red, right? So that red retina will be able to be seen through the almost transparent iris. Now, it's very important to understand that the tyrosinase gene mutation, so the mutated tyrosinase gene, is actually recessive to a normal functioning tyrosinase gene. So that means that if we have a mutated TYR gene and we have a normal TYR gene, that individual will not have the albinism phenotype. And so this means that only in the case that the individual has two TYR mutated genes, so they have both recessive alleles, then albinism will be displayed in the phenotype. So now let's take a look at haemophilia. Haemophilia is a recessive sex-linked disorder. And it happens as a result of a mutation on the F8 gene located on the X chromosome. Okay, so because it's on the X chromosome, it's sex-linked. Now, a normal F8 gene will code for a protein called factor VIII. Now, in Roman numerals, VIII stands for 8. So you know that the F8 gene, factor 8 in Roman numerals. Now, the factor VIII protein, if it's properly functioning, it will, and the enzyme thrombin will then catalyze a reaction which produces the insoluble protein called fibrin. Fibrin is very important in blood clotting. So we can see that without factor VIII, the insoluble protein fibrin will not be produced. And so what then happens in individuals with haemophilia is that when they bleed profusely, they bleed excessively, and this excessive blood loss can be life-threatening. Now, gene technology has enabled us to enable haemophilia-affected individuals to lead a near-normal life. So now let's take a look at sickle cell anemia. Sickle cell anemia is caused because of a mutation on the HBB gene, which is located on chromosome number 11. Now, because it's on chromosome number 11, it's an autosomal mutation, right? Because chromosome pair number 23 are the sex chromosomes. All other chromosomes are autosomes. So the normal HBB gene codes for the B globin polypeptide of a normal hemoglobin molecule, which we refer to as HbA. So the normal hemoglobin molecule is HbA. Now, we know that in a normal hemoglobin molecule, we have two identical alpha globin, so A globin polypeptides, and two identical B globin polypeptides. Now, what happens in sickle cell anemia is that there is a mutation in the HBB gene, which results in valine, in the amino acid valine, being present instead of the normal glutamic acid. Now, this will therefore change the entire structure of the normal hemoglobin molecule, and so we refer to it now as HB, so HBS. Now, what does this mutation actually do, and why is it now going to be HBS? Well, with a mutated B globin polypeptide that now has valine instead of glutamic acid, what's going to happen? So, glutamic acid, the normal amino acid, has hydrophilic R groups, so they love the water. The valine amino acid has hydrophobic R groups, so they are water-hating. Okay, and so what this will do is that they will move away from water. Now, this affects the entire hemoglobin molecule, which is now HBS, because, right, we now have two mutated beta globin polypeptides, right, that both have hydrophobic R groups, and we have those two normal A globin polypeptides. So what I want to say here is we have two that are mutated and two that are normal. Well, that's 50% of the entire hemoglobin molecule, so it will be significantly impacted by this mutation. Okay, and so now we have that HBS molecule. Now, what happens in an HBS molecule is that in low oxygen concentrations, there will be sticky patches of that HBS molecule. And this is where the hemoglobin molecules will begin attaching to one another, and they will form fibers. Now, when they form fibers, that is how we're going to have that sickle cell shape. Red blood cells that have the HBS molecules, the mutated hemoglobin molecules, will have a lifespan of only about 10 to 20 days, compared to that of a normal red blood cell with the normal hemoglobin molecules with a lifespan of 120 days. Now, sickle cell anemia is also an example of codominance, and this is because, so homozygous dominant individuals have both normal HBB genes. They will not be affected. They will be symptomless to sickle cell anemia. Individuals that are homozygous recessive, so they have both of these mutated HBB genes, they will be affected by sickle cell anemia to quite some extent. And then also, where it's an example of codominance is in heterozygous individuals. So individuals that have one normal HBB gene and one mutated HBB gene. Although they will be symptomless in normal conditions, when they start doing exercise and there is a low concentration of oxygen in the body, the HBS molecules will actually start sticking together again, and so that they will reduce the amount of uptake and the amount of transport of oxygen within the body, and so that individual during exercise will feel tired a lot quicker. So sickle cell anemia is a recessive mutation, and it's also an example of codominance. Now, before going any further, let's take a look at the sex chromosomes present in males and females. So females have both XX chromosomes, and males have an X chromosome and a Y chromosome. Now, females, because they have the same two chromosomes, they are referred as the homogametic sex. Males, on the other hand, because they have differing sex chromosomes, they're called the heterogametic sex. Now, Y chromosomes are much smaller than X chromosomes, okay? And let's say this rubber here is our Y chromosome, and the pencil is our X chromosome. Now, we can see here, if these are all genes, there are genes all along these chromosomes. The X chromosome has so many more genes than the Y chromosome. So for example, in this case of haemophilia, it's an F8 gene and it's on the X chromosome. So the male, the Y chromosome, doesn't actually have the F8 gene. So if we have the F8 gene here and it's mutated, although haemophilia is a recessive mutation, if we have that recessive allele on the X chromosome, it will be displayed because there is no other dominant allele to mask the effect of that recessive allele. Okay, so it's very important to understand that the X chromosome is larger than the Y chromosome, and because of that, there are more genes on the X than on the Y, and because of that, a lot of mutations that occur on the X chromosome, even if they are recessive, right, even if they can be masked by a dominant allele, they will probably be expressed because there is no dominant allele on that Y chromosome to mask the effect of that recessive mutation. Now let's take a look at Huntington's disease. Unlike albinism, sickle cell anemia, and haemophilia, which are all recessive mutations, meaning that the mutation that occurs will be masked by a non-mutated gene, Huntington's disease is a dominant mutation. So the mutation occurs on the HTT gene on chromosome number 4. So again, we have an autosome, right? Because it's not chromosome pair number 23, not the sex chromosomes, it's an autosome. And what happens is the HTT gene codes for Huntington, a protein called Huntington. Now, where there is a mutation, glutamine, there will be many repeats of the glutamine amino acid. Now, the more repeats of the glutamine amino acid that there are, the more severe Huntington's disease is. The symptoms of Huntington's disease, well, Huntington's disease results in the progressive deterioration of brain cells, and this will therefore affect motor function, cognitive function, behavioral function, etc. So the more repeats that there are of that glutamine amino acid, the more severe the symptoms of Huntington's disease will be. Now, Huntington's disease, as we discussed, it's a dominant mutation. So okay, there isn't going to be another gene that is going to mask the effect of it as we had in albinism, sickle cell anemia, and haemophilia.